This next one is land-based nutrients and their effect on duration and intensity of red tide events. And this one will be presented by Dr. Miles Medina of the University of Florida Center for Coastal Solutions. The purpose of this presentation is to provide findings from a recent study to identify systematic cause and effect relationships between red tide, in this case Carnea brevis, and anthropogenic drivers. And this relates to the water quality improvement actions of the CCNP, specifically to reduce harmful algal blooms and support HAB research and monitoring and measures to reduce their environmental, social, and economic impacts through the ID and reduction of anthropogenic influences. So with that, Dr. Medina, thanks, over to you.
All right, thanks. Hi, everybody. I'm Miles, and it's a pleasure to be here. Thanks for inviting me to speak with you. I'm a research scientist at the University of Florida Center for Coastal Solutions, and I'll be presenting some of our recent work linking red tide blooms near Charlotte Harbor to nutrients and discharges from the Caloosahatchee River and Lake Okeechobee. First, I want to acknowledge our two co-authors from outside UF. We had Dr. Dave Tomasco from the Sarasota Bay Estuary Program and Dr. Eric Milbrandt at the Sanibel Captiva Conservation Foundation involved in this work. And the research article was recently published in Science of the Total Environment, and I've dropped a link in the chat for free access to the article. Next slide. Okay, so I'm going to start off talking about just some background on Carnea and this little video that shows the data we used for the red tide data that we used for the study. So the video shows Carnea brevis cell counts near Charlotte Harbor from about the last 10 years. And these are data from the NOAA HABSOS database that ultimately came from FWC monitoring. So each frame in the video shows samples collected in the area during a 2-week period. Each dot represents one sample, and the size of each dot corresponds to the measured concentration on a logarithmic scale. So blooms show up as these clusters of red dots, as you see flare up every once in a while. And in terms of the study area, I just want to be clear that when I say Charlotte Harbor or Charlotte Harbor Estuary in this presentation, I'm referring to the broader area from Charlotte Harbor proper, south to the Caloosahatchee Estuary, and then up to a mile out into the Gulf from the coastline. And as the video continues to play, I'll give just a little background on red tide and Carnea. The red tide organism, Carnea brevis, is a type of phytoplankton, and it's not a picky eater. So it uses nutrients in a variety of forms, including both organic and inorganic nitrogen. And of course, these blooms are concerning to us because they're associated with economic costs, particularly to tourism and real estate, as well as public health problems, ecological damage, and wildlife mortality. So the question is, what causes these blooms? Next slide. So first of all, it's well known that these blooms occur naturally in the Gulf, but we've for a long time also suspected that anthropogenic influence or human influence might be important. So research has shown that blooms start offshore due to natural processes, and then ocean currents and circulation patterns can set up a sort of conveyor belt that moves the bloom to the coast under certain conditions. Now, once the blooms are on the coast, they can be maintained and intensified by local nutrient sources, which might include nutrients that ultimately come from our cities and our farms. And it's this last part that my research focuses on. So the big question is whether we're making these blooms worse. And I think it's an important question because on the one hand, we can't do much about the natural causes of blooms. For instance, we're not going to change ocean currents in the Gulf, but we can perhaps do something about our own influence on these blooms if it turns out that we're having a substantial impact on how intense they become or how long they last. Next slide. Okay, so let's dig into this question a little bit deeper. First of all, from earlier research, we know that it is certainly possible that humans are causing these blooms to become more intense. For instance, we know that the K. brevis organism can use the forms of nutrients that we put out onto the coast, nitrogen, phosphorus, and so on. We also know that the amount of nutrients that we're putting out has increased over the past couple centuries as our population has grown and land use has become more intense. And finally, we know that the amount of nutrients that we're putting out can meet a substantial portion of the nutrient requirements of these blooms. On its own, our nutrient output isn't typically enough to sustain a bloom, but it's substantial enough to be a concern. So it's certainly plausible that we could be making blooms worse. Now, going back a couple of decades, researchers have looked a little more directly into this question and into the data to see if there's a link between human activity and blooms. But here the results have been mixed. So on the one hand, there's often a correlation between blooms and river discharge, that is the amount of water coming out of a river. But there hasn't been much evidence of a correlation between blooms and nutrients coming out of those rivers. So is that the end of the story? That is, if we don't see a correlation, does it mean that our impact must be so small that we don't really have to worry about it? And I would argue that the answer is no, not necessarily. It reminds me of the story about a guy looking around for his wallet under a lamppost at night. This is the little cartoon at the bottom. Someone comes along and asks if this is where he lost his wallet, and he says no, but this is where the good light is. And of course, this is a problem that's common to all of science. We're always limited by the information and the data that we have available, and by our ability to ask and answer the right questions. So I would say about this correlation issue that the real question we're concerned with, whether humans are making these blooms worse, is fundamentally a question about causality and not just correlation. So I want to take a minute to just talk about causality and correlation. Next slide. Okay, so we all know that correlation does not necessarily mean causation. Of course, two things can be correlated and completely coincidental and not actually physically related to each other. But what people often don't realize is that the opposite is also true. If you have a lack of correlation, it doesn't necessarily mean that there's no causality. So you can have causality without correlation. And so for our purposes here, it's possible that land-based nutrients help fuel red tide blooms, even though the two are not correlated. And the reason that this is possible is that correlations represent a very simple type of relationship. For instance, a correlation between nutrients and blooms might mean that more nutrients lead to more blooms. But we know that the system's pretty complex. So the interaction between nutrients and blooms might actually be more complicated, or what we call state dependent, which means basically that whether and how nutrients affect blooms can depend on other factors and other conditions being met. What else is going on in the system? The same input of nutrients might have different effects on blooms at different times, depending on what else is going on. And maybe the easiest example, the easiest way to think about this is that if there are no Karenia cells around in the estuary or in the harbor, it doesn't matter how many nutrients you pump in, you're not going to see a Karenia bloom, of course. And then you have to think about all the other, the whole host of factors that come together to produce a bloom. And this combination of factors can determine how strong of an impact land-based nutrients and discharges will have. So fortunately, in this context of complexity, there are methods available for working out causal relationships. These methods are based on the concept of phase space, as pictured in panel B. And this is a concept borrowed from mathematical physics rather than statistics. So it's a very different type of data analysis. The basic idea is that the behavior of a variable over time, for instance, the behavior of Karenia brevis cell counts over time, can tell us about the interactions that occurred during that time. We can visualize the patterns of behavior of different variables as this type of geometric object in panel B. It's called a phase space manifold, and examine the shapes of these objects to learn about causal interactions between variables. So we transform questions about dynamics and causal interactions to questions about geometry, which is much easier to deal with. The important thing to understand here is that this way of analyzing the data automatically respects the complexity of the causal relationships and sort of transcends the limitations that are inherent when you do a correlation analysis. Next slide. Okay, so back in 2020, we published a study using these phase space methods and linking red tide blooms near Charlotte Harbor with nitrogen inputs from the Caloosahatchee River. This was perhaps the first empirical evidence of an anthropogenic link. First of all, we showed that blooms exhibit a strong signal representing a systematic pattern of behavior over time. And the signal is shown in red on the right hand figure. And underneath that, we see the data in black. So these are Karenia cell counts on a log scale, and they show a systematic pattern of behavior. And this systematic pattern of behavior implies that there may be systematic cause and effect relationships at work. And indeed, we found that the link between blooms and nitrogen inputs from the Causality by investigating these phase space manifolds. So these results were pretty novel and interesting. On the one hand, many in the research community and in the public have long suspected this anthropogenic link or even taken it for granted. But the fact is that there hadn't really been very compelling evidence to back it up. Nonetheless, this 2020 paper left a few important questions unanswered, and that's the subject of the study that we've just published. Okay, next slide. So the 2020 study showed us that Causality River nitrogen influences nearby blooms. But we want to know more than just whether it influences it. We want to know, what we really want to know is whether nitrogen is intensifying the blooms. Is it making it worse? And so this is our first hypothesis. We start by determining whether blooms near Charlotte Harbor are consistently being intensified by Causality River discharges at station S79. And on the map, that's that little blue triangle marked S79. And then next, if the answer to this question is yes, then we want to know how much of the watershed is involved in contributing to these blooms. So can we trace the influence further upstream? And basically, we investigate the second hypothesis by walking upstream from the Causality River station S79 to the Lake Okeechobee discharge station S77, which allows water from Lake Okeechobee into the Causality River, and then walk further upstream to the Kissimmee River station S65E that discharges into the lake, looking for causal relationships between each pair of stations. We focused on the Causality River, Lake Okeechobee, Kissimmee River kind of flow path for a few reasons. One is that it's a major flow path to the watershed, but also the flows through the lake and the Causality are managed by the Army Corps, meaning that if these discharges are making blooms worse, then changing the discharge schedule is at least an option. Another reason that we selected this flow path just comes down to data availability. You need a lot of data to run this type of causal analysis, and this area is particularly data rich. Now, there are, of course, other parts of the watershed that we didn't consider in our analysis, and they are important nonetheless, in particular the Peace River, Myakka River, and the coastal areas adjacent to the harbor. Okay, next slide. Okay, so hopefully I gave some helpful intuition about this type of analysis based on this concept of phase space. And I won't spend too much more time on the methodology, but this is a picture of our workflow for analyzing the data. It's this whole cascade of empirical procedures. And to summarize, basically, we start off by taking monitoring data and organizing it into time series to show behavior over time. So these data are the Karenia cell counts, nitrogen concentrations, and so on. And then we recover signals from these time series data. And when I say signal, what I mean is a systematic pattern of behavior over time, like we saw earlier. Next, if we find a strong signal in two variables that interest us, we can construct phase space manifolds and run causal analyses to detect and characterize systematic causal relationships. Okay, so now I'll move on to the results. Next slide. So first, we found that between 2005 and 2021, the Karenia brevis cell counts concentrations showed a strong signal or systematic pattern of behavior. Again, the signal shown in red against the data in black in panel A. And the signal is predominantly seasonal, as you might expect. And panel B shows the phase space manifold. And the important thing to note about the manifold is that it has an organized structure. This reflects the fact that the behavior is structured. There's a pattern of behavior. And a manifold, of course, doesn't necessarily have to show any kind of structure. Sometimes it can just look like a rat's nest, completely disordered. So the fact that this one is structured is what allows us to interrogate it and learn about the causal relationships that give rise to the signal that is shown in red. Okay, next slide. So likewise, we recovered signals from Caloosahatchee River discharge and nutrient data. Here we have discharge. Yep. Here we have discharge, specific conductance, nitrogen, and phosphorus covariates at S79. As you can see, the phase-space manifolds in blue show organized structure that indicates the systematic patterns of behavior. And so we're ready to move on to the causal analysis to see whether and how what's coming out of the Caloosahatchee River affects nearby red tide blooms. Next slide. So looking at data from 2012 to 2021, this is the period where we had like overlapping concurrent data. We found that blooms near Charlotte Harbor were systematically influenced by nitrogen and by discharge from the Caloosahatchee River. And there were some interesting time delays detected in these relationships that I won't have time to get into. So this is the first half of the causality question. Is there a causal relationship to begin with? And on the next slide, which I'll get to in a minute, we'll talk about the nature of that relationship. But first, let me just talk about these results. So like I said, we found influence of nitrogen and discharge from the Caloosahatchee on these blooms. In contrast, we didn't see evidence that phosphorus or specific conductance systematically affected the behavior of these blooms. So our results are consistent with the understanding that blooms are nitrogen limited. That is, in general, we should expect that the behavior of the blooms is governed by the behavior of its limiting factors, and finding that nitrogen dynamics influenced bloom dynamics and that phosphorus dynamics did not influence bloom dynamics suggests nitrogen limitation, at least on this large spatial scale that we analyzed. And finally, it's worth pointing out that this type of analysis included tests to ensure that we're not mistaking mere coincidence for causality. That is, it's always possible that the results are just coincidental, since, for instance, nitrogen and bloom cycles are both seasonal. But the results for nitrogen and discharge both passed this hurdle, and we can conclude that there is a real causal relationship evident here. Okay, next slide. All right, so now going a bit deeper to characterize the nature of this detected causal relationship. The analysis indicates that discharges in nitrogen, both from the Caloosahatchee, both systematically intensify coastal red tide blooms. So in panel A, you can see that both curves, the black and the blue curves, are both positive above zero. The blue curve represents the influence of discharge on blooms over time, and the black curve represents the influence of nitrogen on blooms. So this is a consistent facilitation or intensification kind of relationship. It's always a positive relationship over this time period. Further, we found that while discharge and nitrogen are both consistent in intensifying blooms, the effects vary, and the strength of the effects vary, and they're strongest depending on what stage the bloom is in. So the effect of discharge was typically strongest in the early part of the bloom cycle, and the effect of nitrogen was typically strongest later once blooms were in the maintenance phase. So this implies that water coming out of the river may affect blooms both as a carrier of nitrogen, but also through a separate mechanism. For instance, one of the active hypotheses in our research group, and it's just a hypothesis for now, is that discharges from the Caloosahatchee change circulation patterns around Charlotte Harbor in such a way that they might actually draw red tide cells into the harbor from the Gulf. So that might be part of what starts a local bloom in the harbor. On the other hand, nitrogen seems to play its most important role in maintaining, intensifying, and therefore prolonging blooms. So once the blooms are already going, that's when nitrogen can really sort of shine and play a big role in keeping them going. Okay, so there are definitely some important conclusions here, in particular that nitrogen-enriched discharges have consistently played a role in intensifying and prolonging blooms in this area. And there are also some clues suggesting mechanisms that we're looking into more deeply with our ongoing research. Ultimately, understanding how inputs from the Caloosahatchee River interact with natural processes and translate into blooms should help us figure out what can be done to reduce the severity, duration, and impacts of these blooms on our coast. Okay, next slide. So investigating our first hypothesis showed us that nitrogen inputs from the Caloosahatchee River intensify blooms. And moving on to our second hypothesis, we wanted to know whether this influence can be traced upstream to Lake Okeechobee and to the Kissimmee Basin. That is the source of the problem relatively close to the coast, or does it involve parts of the watershed that are further away? And indeed, the causal tests did indicate contributions from upstream. As you might expect, the nitrogen dynamics at the Caloosahatchee are influenced by nitrogen at Lake Okeechobee, which is influenced by nitrogen at the Kissimmee River. Next slide. To summarize our results, this causal diagram on the left shows the influence of nitrogen from the Kissimmee Basin to the lake, and then to the Caloosahatchee River, where nitrogen inputs and discharges to the coast ultimately intensify red tide blooms. And in case you're not familiar, the notation here is NOx is nitrate plus nitrite, TN is total nitrogen, and Q and the near the bottom is discharge. Okay, next slide. So in terms of takeaways, we found nitrogen-enriched runoff from a vast, highly developed watershed consistently fuels Karenia blooms near Charlotte Harbor. So this, of course, suggests that it's not just coastal areas. The contributions involve the entire watershed. The second point is that correlations don't reliably capture this anthropogenic link. We need a kind of deeper analysis to really bring that out. Third, the timing of Caloosahatchee discharges and nitrogen inputs relative to a bloom's stage of development is important. So it's not just how much, but when. Our results suggest that discharges and nitrogen may act on blooms through distinct but synergistic mechanisms. This is the part I talked about, with discharge having its strongest effect early in a bloom, nitrogen having its strongest effect later. And so mitigating bloom intensity and duration is likely going to require holistic solutions that consider natural drivers, anthropogenic drivers, and their interactions. And we're going to need to look at the entire watershed. Of course, a question I often get is about land use, agriculture versus urban and septic and so on. And with this study, we didn't look, into things resolved at that scale or by land use. We just looked at basically Huc 8 basins. Although, that it is an important question, but we can't really comment on the relative contributions of different land uses and particular subbasins within those Huc 8 boundaries. And then finally, upcoming work from the CCS, Center for Coastal Solutions at UF. We're going to move forward identifying subbasins and land uses that contribute most strongly to blooms. We've got work with the Army Corps modeling alternative Lake Okeechobee operations in order to propose changes to the schedule to reduce coastal impacts. And finally, we've got oceanographers simulating hydrodynamics in 3D to understand how Caloosahatchee discharges affect tidal exchange and circulation patterns in the harbor. All right, next slide. This is my contact info, email, LinkedIn, webpage for CCS. And like I said, I dropped a link for free access to the Science of the Total Environment article there in the chat. And if there's time, I'd be happy to take any questions. Thank you.
Thank you again, Miles, for the presentation. Really interesting stuff there. It's definitely concerning, I guess, to see, well, I guess not concerning, but it's enlightening to see how far the scientists come to be able to, with some degree of certainty, tie back the nutrient loading to these red tide events. So So with that, if anybody has any questions, feel free to pose them now. Then we've got some time. We actually did get a question come in through the chat from Mike Wessel. He states, very interesting work. Can you define what makes the test causal? And if there is a stochastic component, for example, the interaction coefficient. He's also wondering how you dealt with the multi-collinearity in the X maps.
Yeah, okay, it's a good question. So one, yeah, causality is kind of a loose term, so it's worth describing that in a little more detail. When I use causality here in this context, what it means is a like a systematic pattern of interaction over time according to deterministic rules. We can distinguish that, like a pattern of interaction versus another common way of using the term causal, which is like a pulse disturbance. If you get a hurricane or a huge storm or something, and you could say, like the question of Piney Point, for example, you have this kind of pulse event, and then you're talking about what effects did that cause downstream. So that's not what we're talking about. We're talking about a pattern over, nearly a decade, where you see sort of cycles of behavior and cycles of interaction. There's not a stochastic component. This is a more deterministic kind of analysis. Basically, during the signal processing phase, what we're trying to do is filter out what we can call like high dimensional noise that would capture pulse events and things like that. And we're just focusing on the deterministic component, that repeating pattern of behavior. So we're kind of ignoring stochastic, the stochastic component of the dynamics. And then the final question, how you dealt with multi-collinearity. That's not so much an issue with cross-mapping. That's obviously a concern with like statistical methods, like especially regression and so on. Here, this really isn't fundamentally a statistical approach. And so it's fine if things are correlated with each other. It doesn't really present a problem for the analysis.
Thanks, Miles. This is Mike. Yeah, it's really interesting stuff. I guess I'm wondering if you thought about, when you're doing the decomposition of the time series, thought about using the residuals from that decomposition step, where you take the time series pattern and tease out, for example, seasonality out of it, and then use the residuals of that to kind of try and map that to other things, like nutrient concentrations or in your lags or something like that. I'm just curious if that's the path that you took, or if you thought about taking that path and or not, or did something else.
Yeah, it's interesting because, okay, so for the Karenia signal in this case accounted for 2/3 of the variance, which means there's still a third on the table that we're not even dealing with. And so there are certainly things you can do with those residuals. One thing that I've done before, not for this analysis, but one thing you can do is just look at sort of extreme value analysis, and talk about like return periods and magnitudes for for how often some extreme value might be observed. And there's really all kinds of things you can do. I mean, you can run a lot of different statistical things on the residuals. It's really just motivated by whatever the hypothesis is. Here, our focus was on the deterministic or systematic component because I think that's what's really relevant when it comes to policy. Is if like it would be one thing to say we made the 2018 bloom worse, right? And it was a one-off. It doesn't usually happen, but like in that case, it did, right? And, if that was the result, there might be certain policy implications for that. But if we say instead, we ask instead, are we having a systematic effect over time? Is it consistent? I think that's pretty compelling. So that was our focus here. We don't get into like, we affected it this much in this year versus that year and so on. Although, that could be interesting as well. So
Yeah.
I think there's more that could be done in that direction for sure. But right, it wasn't our focus for the study.
Yeah, well, it's very cool. I mean, I think these new methods definitely are very much needed in this regard to explain really complex phenomenon like this. So nice presentation. I appreciate it.
Cool, thank you.
So we've got a couple of questions come in. One from Chris Anastasio. So yes, if there's a way to take the results coupled with rainfall or other external factors and develop forecast models for the Cornea brevis.
Yeah. Hey, Chris. Thanks for the question. Good to see you. Yeah, I think that's interesting. I mean, I'm more on the like inference side, like looking back and seeing, what happened. But we have computer scientists and people that are interested in forecasting. And so I guess, one thing is just like establishing that what comes off the land is important. Means that you have to consider that if you're going to do forecasting. And ultimately it is, driven by rainfall. So I think ultimately that's something our group is interested in working towards, but we're not quite there yet. We're building towards it.
Hey, Miles, I had a quick follow on. And again, I think I was commenting, this is like the third time I've heard your talk. I learn something every time I hear it. So this is awesome work. One thing I was just curious as a follow, not really a follow-up, just another question is that if you were like king of the world and you could select any data that you could use to help better these relationships, I guess my question is, are we collecting the right data to drive to continue to drive these relationships? Or are there other types of data that we should be maybe looking at in addition to what's already being collected?
Yeah, I mean, that's always the limitation. If you have any suggestions, I'd be glad to hear them. I mean, what my mind goes to
That's what I'm
automatically.
asking you.
What I go to automatically is like, we could look at the Caloosahatchee and the lake and so on because those are so well monitored, particularly because the flows are managed. But there's other basins there, Peace, Mayaka, and so on, and so those aren't as well monitored because they're not, there aren't structures there. So I think having more frequent and more consistent data would enable this kind of analysis over a wider area, include more basins in it. I mean, there's other ways we can approach it within the constraints of the data limitations, but if I was, if I was king of the world, I think, I'd want to have as much nutrient concentration data and flow data as spatially resolved as possible.
And I'm not really asking more. This is as much a hypothetical question as a real question. I don't know if Brandon Moody's still at the meeting, but he may be a good guy to talk to a little bit about that too. Now he's Charlotte County,
yeah.
But he could, he's looking at developing additional monitoring sites, and that might be really cool for you guys to talk if you haven't already, and see if there's some ideas that you guys could share and potentially collect some of those additional data.
Yeah, totally. We are in touch with him.
Cool.
He's come to us a few times to get our view on stuff and our suggestions, because it seems like he's designing a really cool program to supplement collection efforts that are already out there.
Good job, man.
Cool, thanks.
So I think we have time for one more question. Mark Walton, did you have something you'd like to add?
I'll get mess you up. I've got about 3 questions I was going to ask. Miles, great work, and I'm just going to take a while to wrap my head around, to be completely honest. But the initial space where you look at the geometry of the relationships, how do you determine the strength of that causality or or relationship? Is there a metric that goes into that, or is it just an eyeball? You sort of see how tight the curves are, or how?
Yeah. So you can often see it visually, but there is an algorithm, fairly simple and a metric. So basically, let's see, can we go back to slide, I don't know, slide 10 maybe? Um, oh, sorry, too far. The one called signal recovery. I think it's like 2 or 3 slides down. Okay, this one will work. Okay, so right, so basically the causality test, the first one that just tells us if there is a relationship. What we do is we can, let's say there's the 2 variables we're interested in are the cornea, like we're seeing here, and then also like total nitrogen out of S79, which is one of the ones that we found a positive result for. What we do is we use the signal to reconstruct this phase space manifold, and it has a structure to it. We do the same thing in parallel for the other variable, total nitrogen, for example. And the question is whether there's a sort of correspondence in their, I say geometry, but technically it's topology, like sort of in their shape. And the way that you test that is through this procedure called convergent cross mapping, which is actually quite simple. You basically pick a point on one manifold, look at it, look in the neighborhood of that point. So other points that are close to it in this space. Advance those points one step forward and take a sort of weighted average of their positions. And then that gives you a prediction for what the point should be at the next time step. And well, I skipped something. So you get, I don't know, I guess kind of technical, but you're basically trying to use the time indices on one to predict points on the other. So there's a kind of prediction scheme going on, and you just carry that through the entire structure, so all the different time periods. And you start off with a very small, what's called library or base for prediction. So you actually ignore a lot of the points at first, and then you iteratively increase the prediction base until you're using the entire manifold. And what you see that gives a positive result is that as you increase the prediction base, the number of points you use for prediction, the prediction skill increases and gradually like kind of just converges and sort of plateaus to some level. And that prediction skill is just measured as the Pearson correlation between the predicted value and the actual value on the target attractor that you're predicting. So yeah, that's pretty much it. So there's this kind of prediction across attractors, and then you just look at the correlation between the predictions and the actual.
Groundbreaking. Devin, have I got time for a couple of other ones, or at least one more?
Yeah, we can do one more, and then we're going to try and do a short break, like 5 or 10 minutes. So.
Okay. You mentioned the signal for, I think the discharge was important early in the bloom, and then the nutrient signal sort of picked up towards the end of the bloom. Mm-hmm. You mentioned the potentially sucking in Gulf water due to the circulation. Is there any other, I guess, narrative to that? Is salinity an issue in the early part? Was that signal, I guess, for the discharge a negative impact on the counts? I wonder if you just sort of elaborate on what you think might be going on with those time lags.
Yeah, it's a great question. It really took some thinking to figure out how to interpret that. Oh, one more note, if you're interested in the convergent cross mapping, the causality method, that's a paper, I think it's open access by Sugihara from 2012. So worth checking out. It's a good read. It's pretty short. I think it was published in Science. But yeah, to your question about the discharges. So from the start, I mean, we hypothesized that the discharges would have a consistent intensification effect, but we did in the back of our heads think, well, maybe it's not necessarily the case, right? Maybe sometimes it's a positive effect and sometimes it's negative because, as you mentioned, if you have a big flush of low salinity freshwater, that might actually inhibit the blooms, right? So it wasn't necessarily a given that we would see the positive result that we did. We hypothesized it, but we knew that like maybe it varies, and that would be interesting. So I think the other major interpretation for a consistent positive effect that that is strongest in the early part of the bloom is, you know, maybe that discharge is a proxy for some other nutrients, iron or silica or something, that helps get the bloom going. So, you know, it could just be about water quality, it might be about circulation, and it kind of points towards different possibilities. The results point towards different possibilities. But, you know, we can only speculate on that basis.
Great, thanks.
Thanks again, Miles. Really great and interesting research there. Looking forward to seeing kind of what you guys do in the future on that. So
cool. Thanks a lot, everybody.
Yeah.